everyone, and thank you for accepting this oral presentation. So I present myself, I'm Dr. Wara Beliza, and I am a senior lecturer at the University of Béjaïa and attached to the Laboratory of Applied Microbiology. So I'm here today to talk about a specific topic, which is the candidiasis infections and palm wine as a source of probiotic lactic acid bacteria with antifungal properties. So to start, let's introduce this source, which is palm wine. So palm wine is a traditional beverage that we can find in Asia, but also in some countries in Africa. We can also find it in the clandestine way in some region of the south of Algeria, like Ouedsou, for example. And here we call it the legmi. In general, in Algeria, we call it legmi and not the palm wine. So palm wine is rich in sugar and nutrients because it is a beverage that you can obtain by the fermentation of the palm directly. So it is commonly consumed in tropical and subtropical regions, and it is valued not only because it is a refreshing drink, but because it has also health benefits sometimes, because it is rich in nutrients like magnesium and some acid, which is beneficial to the gut. So here our topic will be more about finding a solution using this palm wine as a source for probiotics. So our public health issue here is vaginal candidiasis. Vaginal candidiasis is a really common infection that attends women, and it is caused by the yeast Candida albicans. So here the issue is because it is really difficult to cure this kind of disease because there is a problem of recurrence and antifungal resistance. So most of women has at least one episode of this type of infection during her life because it appears when the balance of the vaginal microbiota is disturbed. This can occur while you use antibiotics or some other hygienic, for example, perturbants, etc., or hormonal or sexual life. So here, why using palm wine? Here we saw some publications which were talking about the adaptation of some strains when you use them on another microorganism ecosystem. Here, for example, this study showed that the probiotic properties of lactic acid bacteria taken from palm wine can be adapted to the gut microbiota. So we said and we thought that why not trying to use them into the vaginal microbiota. Here we see also that the palm wine has an acidic pH like the vaginal microbiota, so the adaptation will be easily conducted. This is why we saw it as an opportunity for our isolation. So the objective of our study is, first of all, isolate and identify lactic acid bacteria, and also trying to test their probiotic traits in order to evaluate their antifungal properties against Candida albicans from the vaginal microsystem. For the sampling, so here we used 2 palm wine samples only. It has been provided from a colleague from the region of Ouedsou in Algeria between February and March 2025, and directly conducted into the laboratory in order to make some assay. For the isolation, we used the mandragosa and sharp broth and agar, which is used generally in order to have lactic acid bacteria and also Lactobacillus genera. This is our first aim. And we used a preliminary identification, which I mean gram staining and catalase test in order to really purchase this type of strains. Then we started with the safety, of course, to see if we can adapt these strains into our vaginal ecosystem. So there was the hemolytic activity using blood agar, and then some tests to see the property of the surfaces, like hydrophobicity and the co-aggregation, and also auto-aggregation. Also, we conducted some tests to see if our strains were able to form biofilm. Here we have to know also that biofilm is really important. Either you are working on beneficial strains or pathogenic strains. If it is a beneficial strains, it will be more important because if your strain can attach to a mucosa or a surface, it means it will remain. And if it is pathogenic, it means that the virulence is higher than you think. So here we conducted the test using the staining of crystal violet, using microplates and absorbers to see the results. For the strains tested on, I mean the targets, we used 7 clinical strains obtained from women with infections here in Béjaïa. And the characteristic of these strains are that they are all hemolytic, all resistant to the common antifungal agents like fluconazole. And also, they are biofilm forming, high biofilm forming ability. So, which means that these strains are very virulent. For the evaluation of the antagonistic power, we use the well-known spot test, which means we put directly the strain that we want to test and the target on the same agar plate, and we try directly to make a grow and see if there is an effect. Also, we have conducted a test to see the anti-biofilm power. So we saw first we wanted to know if our strains form biofilm, and also we wanted to know here if our strain can erase a biofilm or make a kind of a competition between the pathogenic strains in order to not let them form this biofilm on the vaginal mucosa. For our results, so we based our result on the phenotypic way. First of it was the growth on Manragozza and Sharp agar. Here we obtained colonies which reveal directly that it is lactic acid bacteria, and also gram-positive and negative catalase reaction to H2O2, which means that we are on the good way to see if it is a lactobacillus. For the hemolysis, all our strains, so here we isolated 5 strains in total, and all of them were no hemolysis. There is no hemolysis on the test, which means that we can use them safely. For the cell surface hydrophobicity, so we had pretty high hydrophobicity ranging from 95% to 100% sometimes, which here give us information about our strains. It means that this hydrophobicity can lead to easily a formation of biofilm in the vaginal mucosa. Also for the antagonism, here for our strains, like noted VP4, VP6, we can see that we have a notable antifungal activity using this spot test with inhibition zone going into 20 millimeters against certain Candida albicans strain. So here, if we take this one and the biofilm coaggregation formation, we can really lead to a good probiotic. Same thing here for the biofilm formation. So here we see that we have moderate biofilm formation and a strong inhibition. Here we see the biofilm formation and the inhibition of the formation of Candida albicans. So here we have 7 strains tested for all, and we see that we have a strong inhibition, 60% to 90% inhibition of the adhesion of Candida albicans. Here it means that we have a real competition, even though for the antagonistic test we didn't really obtain big zones of inhibition. But here there is another indicator, which is the biofilm. So even though the inhibition is not really important, there can be another mechanism, which is the anti-biofilm formation and the co-aggregation, which indicate promising probiotic potential. Here it is the same for co-aggregation. As we see here, we have good rates for anti-inhibition. Here we have also good rates for some strains in co-aggregation, going into 70% with some strains of Candida albicans, which really is important to note. To conclude, I can say that palm wine is an interesting source of probiotic acid lactic bacteria, and it can lead really to isolate and obtain some strains that you can use in the human system. Although the vaginal microbiota is not well studied and there are not really much researches about it, because in general we prefer study gut microbiota, but there is really a pretty high interference in this way. So our results suggest a promising potential for preventing vaginal candidiasis and also to cure them. And further studies, of course, have to be conducted in order to see the molecular identification and some in vivo evaluation in order to conclude this study. I finish my presentation with thank you for all the persons that have made a valuable contribution, starting with Professor Faraji, which is here, and which is the head of our team on the Laboratory of Applied Microbiology. And thank you for your attention.